I first off would like to thank you, Dr. Sondheimer, for taking the time to meet us tonight. I know that things are probably quite busy over at Synlogic with everything changing recently. So thank you for coming in. And I'm just going to ask first that for those that are still kind of hearing about the trial maybe for the first time, if you could just let us know in a few sentences the concept that was being trialed.
Sure. So the drug that we were working on, which was known as either SYNPHENY 1934 or lavafenagine marsilicobacter, was a very new type of medication called a synthetic biotic. What a synthetic biotic is a living bacteria that has been engineered to do specific jobs. The entire concept of our company is built around the idea of using synthetic biotics to build new medicines. And what we can do with them is insert genes into the bacteria that, in the case of PKU, allows it to take up phenylalanine from food that you eat or from another pathway where phenylalanine circulates out of your blood and back into your intestine, and then degrade that phenylalanine using the enzyme phenylalanine ammonia lyase, which is the same enzyme that's in the drug PALYNZIQ, which is approved in the US and recently in Canada. And so that's the concept of the medication, is that had the medication been effective, it would have allowed people with PKU to greatly increase the amount of phenylalanine they could tolerate, because it would basically remove the phenylalanine from your food before you absorbed it. The drug was through what's called phase 2 of its study. And so phase 2 is when we tried it in people who had PKU for the first time. And we were in the middle of the phase 3 study, which is the pivotal study. It's the last study that we need to do prior to the approval of the drug, up until about 2 weeks ago.
And we all know, or will know with this, that the trial did end earlier than expected. Can you let us know why this happened?
Sure. So at specific points in the trial, we look at the data to try to understand if there's sufficient benefit to people taking the drug that's in good trade-off against the risk of taking it. It's important to come up with drugs that are more beneficial than they are harmful. And all drugs have side effects. We can see the side effect profile during the trial at any time. As we were running the study, I would continuously get information about side effects that people were experiencing. So I knew those. But we only look at specific predetermined points at information about how well the drug is working. We were approaching one of those points when we had to present our data to a group of outside experts known as a Data Safety Monitoring Committee, or DMC. And in order to prepare for that meeting, we produced a test set of data allowing us to look to make sure that when we actually gave it to the DMC, that it worked properly, that it didn't have any errors. When we looked at that data, we realized that there was a problem with the potency of the medication, that effectively it was not lowering phenylalanine at any of the doses that we were given. When we saw that, we were obligated essentially to stop the trial. We can't work on a medication that doesn't appear to be effective in treating PKU. And that's sort of a principle of human subjects research. When we do human subjects research, people who volunteer to be in those studies are taking a lot of risks. They're using something that hasn't been used before, or hasn't been used for as long. And one of the things that they are owed is someone safeguarding them. against something that either doesn't work or is dangerous. And the whole purpose of the architecture that looks over the trial is to make sure that risk and benefits stay in balance for participants in the trial. When we saw the data, we decided that even though our risk profile was actually better than expected, the benefit profile wasn't good enough to support it. And that meant that we had to stop the trial.
Now, there was never a safety issue for people on the trial.
No, there was not. And in fact, the safety data looked reasonably better than in the phase 2 study. During our phase 2 study, we had too many participants drop out of the study because they were experiencing adverse events. Folks who stayed in the study had lowering of their phenylalanine. And we thought we understood how to improve the experience of taking the medication. We made a bunch of changes to the way the medication was delivered to people. And those changes did reduce the troublesome side effects that we saw in the phase 2. We had many fewer people leave the study. So we had 37 people in the study at the time we stopped it. And at that time, only 3 people had discontinued from the study. In the phase 2, which was only 2 weeks of length, so the phase 3 study was 3 years, almost half of the patients had discontinued before 2 weeks had come. So we had done much better on safety. We didn't have what are known as serious adverse events. Serious adverse events you can think of as something that causes you to be hospitalized. Is the easiest way to look at it. So anything that's causing you to be hospitalized or is worse than that is generally regarded as a serious adverse event. So that happened in the study. There were things that we were particularly looking out for in the study. So we were looking out for people having problems with allergic reactions. And that's because of the history of phenylalanine ammonium lyase PALYNZIQ. So because it's an allergen for that medication, we wanted to be sure that we weren't having allergic reactions. We were, of course, concerned about infections because we are using a bacteria. So we wanted to be sure there was no evidence of any infection. And we didn't see any signs of infections or allergic reactions. So it was not stopped because of safety. If there had been a safety problem, we actually wouldn't have needed to look at new data again because we see safety data all the time. The study actually had many pre-specified stopping criteria. So if anyone had an infection or an allergic reaction, we actually would have had to at least freeze enrollment. And if too many bad things happened, we would have had to stop the study. But there was never any issue that suggested to stop in terms of safety.
Now, when looking at it, because we know that phase 2 was working at least at one point, was there any way to increase the dosage to obtain better results, or maybe a longer use of the medication would have eventually lowered these Phe levels? Was it acts too soon, I guess is.
Yes. So based on phase 2, we were using doses that appeared to be appropriate. So we were really using the same doses in phase 3 as in phase 2. We were just getting to them more slowly. There are some wrinkles in the phase 2 study. On the very last day, participants took a dose that was 2 times as high as the dose that they were taking 3 times a day in the SYNPHENY study. Now, the total dose per day in the SYNPHENY study was high because you were getting it 3 times a day. And it was lower in the phase 2. But I don't think it was an issue of dose. And one of the reasons that we believe that to be the case is that the response for phenylalanine level was evaluated at all 3 of our dose levels. And there was no trend that phenylalanine was lower at higher doses. it was essentially flat. And so if it was flat over that range, there was no clear reason to expect that higher doses would have been efficacious. And there is a limit to how much of a live biotherapeutic you can give. The medicine was given as a sachet that was dissolved in liquid. And we were pretty much up against the amount that we could put into 100 mLs of liquid, which is about 3 ounces, something people can conveniently drink after a meal. Had we increased the dose, we would have had to scale that beverage up proportionally. And there are limits to what we can ask participants to do. We can't ask them to eat 10 grams of a live biotherapeutic. That's not going to work.
Fair. Those who participated in the trial, we've had some asking, will they ever know what their own Phe results were at the time of the trial stopping?
So they won't, and that's for complicated reasons. So first of all, we don't know who they are. So we don't know who any of the participants in the trial were. That's a requirement. We are blinded through the investigators who were at the local sites and actually running the study. So those folks knew who was in the study, but we did not. They didn't tell us who they were. We were all blinded to the Phe values in the study, and we simply got them as numbers. And so we can't really detangle them and provide them back. And that was something that we told participants during the study. Now, there was what's called an open label extension. And the open label extension, a couple of Canadian patients had made it into. I don't know if they had Phe values for an open label extension. That would have taken 3 months in open label extension. And I'm struggling right now to remember whether the longest Canadian patient would have hit that point. But I think the answer is
so even if the trial hadn't been stopped early, this information still would have been blinded.
well, the open label extension data
other than open label.
Yeah. So there were 3 phases of the study. There was an escalation part that lasted 9 weeks for most participants. There was a 4-week period where they received either placebo or the active product. And then there were 3 years of open label extension. Now, people weren't barred from having their Phe levels checked by their usual means. So people were able to have their Phe tested at the time. We just couldn't be told what those values.
Fair. So is this idea or concept of what was commonly known as a gut bacteria protein to eat up the Phe, is this idea gone forever, or might it need some tweaking? Is that tweaking a Synlogic project or maybe someone else? Where is this going, do you think?
So that's a great question. And, we've been struggling to try to understand this. This was obviously a shock to us. We didn't expect these results. We don't know why the drug didn't work. I think the 2 major theories that I have are there was something different about the drug that we produced for the phase 3 study from the phase 2 study that allowed the drug to work in phase 2, but not in phase 3. And what that is, we're not sure. One of the problems is we're the first company to try to make a synthetic bio and use it in participants. So when we run into problems, we're the first one to ever run in. We wish we could solve that issue if it was a product issue. What we know is the way we test medicines at Synlogic when we're not giving them to patients is through a test that's done in a test tube. We basically give the bacteria phenylalanine, and we see that it produces metabolites from Phe. So that works in our phase 3 product. The problem is a test tube and a human are not the same. And the only way to find out if a drug works in humans is to give it to them, which is what we were doing. This was effectively the test to see if it was going to be effective. We had to change a lot of things about the way we made the drug, mostly because of scale. When we made the drug for the phase 2 study, we were providing 10 patients with drug for 2 weeks. In this study, we were providing 150 patients with drug for 3 years. So it's many orders of magnitude more drug. And that meant we had to make changes. We couldn't make the drug the way we made it in phase 2. We would have never made it. We had to use larger fermenters. We had to use machines to fill the sachets rather than filling them by hand and weighing them, because that's something you have to do when you make that many doses of drug. We changed things that there was no reason to think would have impacted the way the drug worked. People did not like the flavoring agent that was used in phase 2, so we changed it. There's no reason that flavoring agent should have done anything, but we don't know.
The magic of a trial, right, is to figure these things out, kind of.
That's the purpose of a trial. Trial, unfortunately, is an experiment to see if something works, and experiments are complicated. And in this case, the experiment didn't work. There's another possibility, which is that there's something about the interaction of people with probiotics that we simply didn't know before the study. So people have used probiotics for forever. So people take probiotics for gut health. But when they're taking them, when you take bacteria and eat them, the part of your body that the bacteria is growing is the lowest part of your intestine, called the colon, because that's where bacteria live. But that's not where SYNPHENY 1934 has to work. It actually has to work way up at the top, right after the stomach, because that's where you absorb phenylalanine from food. So SYNPHENY 1934, although it was a living bacteria, it couldn't divide. And so one of the issues that's possible is that it doesn't make any difference if you're taking probiotics for gut health. If your stomach kills 99% of the bacteria can regrow. Bacteria divide very, very rapidly. So you wouldn't even notice if you developed a defense mechanism against eating bacteria that was killing. But for SYNPHENY 1934, which can't divide, and it has to work in the upper part of the small intestine, if people over the course of a few weeks developed a resistance, that would actually destroy the ability for our product to work. Since nobody's ever tested a non-dividing probiotic, we're kind of in this new space where it's possible that there's a human defense mechanism against this, and no one had uncovered it.
Because they were only on it for a couple of weeks in phase 2.
In phase 2, people were only on it for a couple of weeks. Now, this isn't the likeliest idea, and it's not something that we didn't think about, but we didn't believe it was likely that there was a mechanism that wouldn't be seen in 2 weeks, but would be seen in 12 weeks. That seemed implausible to us. And, effectively, the only way to find out, again, is to study humans. That's the only way to know.
So the PKU community has been pretty fortunate with KUVAN and PALYNZIQ through their trials. They went to market after trial, so we've seen a lot of positive outcomes from trials. And I mean, there's other companies. I know PTC has done a trial for PKU recently as well. So, but these results are not common for a rare disease space to have this many drugs come out of trial successfully. Do you know what trials look like on average without positive results to report, like those that have to stop early or don't?
So most drugs that are developed fail. There's actually an unfortunately named concept called the Valley of Death, which is where drugs fall into and then they die. They don't work for you. That valley of death is most severe at earlier stages. So people come up with many, many ideas for drugs and try to make them work. And most of them fail in preclinical studies or the first time they're given to patients. Actually, that's not quite right. The first time they're given to healthy volunteers in a phase 1 study. That's where most drugs actually fail. Phase 2 failures, when you give them to people who actually have the disorder, are less common than the earlier stage failures. And phase 3 failures are less common than that. So it is a reasonable proposition when you go into a phase 3 that the drug is going. You're doing it, frankly, because you think you have to show the drugs work. So it's more common than not that phase 3 trials don't fail, but they do from time to time. Recently there was a gigantic study of Alzheimer's that effectively failed, that did not meet its primary endpoints. And this was a huge study. I don't know how many thousands of people were in. So it happens. It doesn't happen a lot, but it does happen.
Fair. For many people in the community, this may have been their first step or their first opportunity at being involved in a trial and to get really excited about one. I know there's a lot of excitement at our events around this one. So this is being felt as a really tough time for sure, with people reaching out to us in frustration and disappointment or disbelief. What can be said to them about trial participation as they're going through this stage and looking at the next, perhaps?
So it is hard. It is hard for participants, and I know that. Again, as you know, I'm a doctor who takes care of people with PKU and other metabolic disorders. And this is not the first time I've been involved in a trial that hasn't worked. I've been an investigator at studies that have not worked. And it's hard. People volunteer to be in trials, and they do it because they think it's going to help them with the disease they have. And they place their emotional chips on this working. And there's a well-known effect of actually being in a trial. People feel better about their health just by participating in the trial. You see this even when people, this is why we give people placebo, actually, is because this effect is so strong that we have to give people fake drug to pull apart how they feel about participating in the trial from how the drug is. So it is difficult. It also felt very, very abrupt for people. Unfortunately, the rules for how you shut down a trial are somewhat inhumane. There is a reason for that, but it still doesn't feel great. We didn't know who anyone in the study was, so there was no way for us to reach out to people and tell them, hey, we're stopping the study. We also were not allowed to tell the trial sites that we were going to close the study in advance. And the reason we weren't allowed to do that, unfortunately, is a legal and financial one. So we are a publicly traded company. And when something happens, we have a window in which we have to disclose information. We're legally required to do that. And we have to do it through a press release, unfortunately, so that everybody in the world can see it at the same time. So what we did is on February 8th, we issued a press release at 4:50 in the afternoon. And then we immediately sent letters to every trial investigator and tried to get on the phone, find these people to tell them to read the book we wrote so that they could get in touch with their patients. Now, in a world that's not connected on the internet, this can work because people don't read the financial news. But in a uh, issuing a press release, everybody with PKU is going to know it has happened far sooner than the investment. I would guess of the 37 people in the study that nearly all of them, unfortunately, are through and not through investigating. And it's very unfortunate, but we were required to do it. We could not tell people what was happening. We had to act very fast. It was also important for us to act very fast because the key thing for me, the company, was protecting the health of people with PKU. When we knew that the drug was not going to work, we had to stop the trial. We couldn't continue. And it feels cruel that people will think, oh my gosh, I think this medication is working. But unfortunately, we have to look at the data from everything, and we can see that it's not working. And if it's not working, we can't continue the trial. One of the things that we promise the people who are going to be in trial is we protect that risk benefit. That we look to see what bad things can happen to people and what good things can happen. And we have to be sure that the good outweighs the bad. If it doesn't, it's not a drug that people should be taking. And unfortunately, ours was not a drug people should take. And so we had to stop.
As bottom line is, we want our fee reduced.
Everybody wants their fee number better. They want it easier to control. We wanted that too. And if our drug wasn't going to do that, then it wasn't.
And people need to know, yeah.
Wasn't going to be a drug that anybody could use. And so we did this because we couldn't take risks with people with PKU for something that wasn't.
Now, we understand that Canada had 4 active sites in Canada, already up and running, active at the time the decision was made to end the trial. And we're pretty proud of the fact that we did hear that some of these sites were most active around the globe, which is pretty exciting for Canadians.
Yep.
And so what does this say to other pharmaceutical companies who may be looking at, is Canada worth it? Because we don't see as many trials as other countries do.
So,
amend to that. I know you also corrected me. There was 6 registered.
There were actually 6 sites that were planning to participate. There were 4 sites that were active at the time the study ended. Those were Dr. Pond's site in Calgary, Dr. Potter's site in Hamilton, Dr. Nemo's site in Toronto, Dr. Bork's site in Ottawa. And we were planning to open sites in Vancouver, Montreal, but those sites haven't reached full activation. For those who are listening who don't know me, I'm at Sick Kids myself, so and I'm a Canadian. So I think maybe part of the reason that Canada outperformed here is that I'm from this community. Synlogic is not a Canadian, I'm just a Canadian worker. But it's certainly true that of traditional sites, the top 2 traditional sites in the study were in Canada. They were the site in Calgary, the site, and they were fantastic partners. I think there are many reasons for that. We actually, I should say, also had a phase 2 study. We had Canadians travel to the U.S. to be in the study. I think it's partially, frankly, due to CanPKU. Promoting people's involvement in trials. I think that's helpful. I think it is also true that Canada is a surprisingly easy place to do clinical studies. We don't have some of the hurdles that U.S. academic health centers do. And I certainly hope that more companies will be interested in doing studies in Canada. I'm glad that PTC has done studies of their drug in Canada, and I believe they're going to continue to do that. I hope that Janana, who has a drug that's completed phase 1 and is looking to do later phase studies for PKU, will also do studies in Canada. I think it's a good place to work with a very reasonable regulatory environment. I don't want to say bad things about the US Food and Drug Administration, but I would take working with Health Canada any day of the week over working with the FDA. They are simply much more reasonable. They're much easier to talk to. They actually, I don't want to say the FDA doesn't care about patients, but I kind of do.
You don't want to get you in trouble either. We need you.
Health Canada really does, and does a better job of just patients and to talking to people who do drug development.
Okay, thank you. Again, I want to my heart broke for PKU people as much as it did the Synlogic team, because we have had such a good working relationship with you guys. And I know heart and soul went into this for everyone. So, so I really want to thank Synlogic for their sponsorship, their active attendance at our events, for including Canada and investing in us as a country, as well as us as patients, with doing this trial. And thank you so much today for being here. It's really good that you're speaking to the community and letting them know
Sure.
what's between the lines of a press release, because we don't always.
And the one message I'd like to leave people with who have PKU in Canada or participated in the trial is not to come away from this experience deciding never to be involved in research on PKU again. Please don't do that. You should be in studies by PTC and Janana. These companies, like our companies, are trying to develop drugs that will help you. And what you should take away from this is not that, our drug failed and this was a terrible experience, and how can a company treat you this way? But I'd like you to view it as, we tried something, tried to make drugs for PKU. We all really cared about PKU. We didn't succeed, and when we saw we weren't succeeding, we stopped. And we stopped because we didn't want to take any risk with your health. We didn't want to waste your time. And we also wanted to clear the field for other people who are working in drug development on PKU. It's very, very important that we do. To monopolize the clinical development space when you have a drug that doesn't work is simply not fair. So we needed to get out of the way. Most people listening know that Synlogic will actually cease operations within the next couple of months because we needed this drug to succeed. It's very sad. We had a program for homocystinuria as well that will now not reach patients. We hope that someday someone may solve the problems that we're able to and be able to resurrect these drugs. I don't know if that will happen. But please don't give up on research just because this did not work.
Yeah, if we fail, we try and try again, right? Yeah, we really appreciate it. And I thank you for your time and taking this.